Good day everyone! Today I will explain how long-term memory is achieved using the organism Afflesia californica, a marine sea slug commonly used in neuroscience research. Afflesia is ideal for studying memory because it has simple neural circuits that allows us to clearly observe how learning happens at the cellular level. Particularly, scientists usually study sensitization using this organism, where a harmless stimulus becomes stronger after repeated shocks, and how this leads to long-term facilitation at the synapse. But before everything else, this video demonstration is supported by the study of Randolph and Fever of 2023 entitled Improvements in Operant Memory of Afflesia are correlated with age and specific gene expression. Their study explains that memory formation involves gene activation that leads to physical changes at the synapses, and certain genes may make learning more effective. Using young and aged Afflesia, the study found that aged animals who were able to learn showed better performance and had higher baseline levels of APC and EBP and tenure in mRNA before training. These elevated gene expression levels may have primed their neurons for synaptic change, enhancing learning ability in aged animals that successfully acquired the task. In Afflesia, long-term memory is studied through a process called sensitization, when the animal experiences repeated or strong stimulation in the sensory neuron. Its response to a mild touch becomes stronger and lasts for days or even weeks. This lasting behavioral change indicates long-term memory. The neural circuit is very simple. A sensory neuron detects the stimulus and communicates with a motor neuron, which produces the withdrawal response. When the sensory neuron is activated repeatedly, it releases a neurotransmitter called serotonin onto the motor neuron. When serotonin binds to the receptors onto the motor neuron, it activates a cell signaling pathway inside the cell. This process is called signal transduction, where an external chemical signal is converted into internal molecular events. The signaling cascade continues when CAMP activates protein kinase A or PKA. For long-term memory, PKA moves into the nucleus and activates a transcription factor called CAMP response element binding protein or CREB, which turns on memory-related genes. Once CREB is activated, gene transcription occurs, producing new proteins. These proteins are necessary to strengthen synaptic connections and is also critical for converting short-term changes into long-term memory. The newly synthesized proteins are transported back to the synapse, where they cause structural changes. This includes the formation of new synaptic connections and strengthening of existing ones, which allows the memory to persist. In conclusion, long-term memory in Afflesia shows how simple experiences can lead to lasting biological change. Through cell signaling and signal transduction, a brief stimulus triggers serotonin release, activates intracellular pathways, induces gene transcription, and results in structural synaptic modifications. These molecular and cellular events transform short-term neural activity into long-term memory. By understanding this process in Afflesia, we gain insight into the fundamental mechanisms of learning and memory in more complex organisms, including humans. And that's all for long-term memory in Afflesia. I hope you've gained something, and see you next time!